Good morning, everybody. My name is NCotter, and with the help of the St. Leo University Mathematics and Science Department and by the supervision of my mentor, Dr. Darren Bell, I did my research on the microcidal effects of linoleic and palmitic acid synergy applied to, specifically, Staphylococcus aureus. I chose these two acids specifically because they showed synergy with other organic fatty acids and by themselves proved to be minimally effective against Staph aureus bacteria. Now, why I chose organic fatty acids specifically is that they are non-toxic, they're very easily extracted, and they're very affordable. These acids can be found in a wide range of any sort of organism, or, for instance, palmitic acid is a derivative of plant roots, and linoleic acid can be found in areas as ubiquitous as skin cells. So, originally I had intended to use cybaric acid, but because cybaric acid, although it is easily extracted, it comes from a mushroom called Cantharellus cybarius, or the chanterelle mushroom, there's a patent pending on the pure form of this acid. So, although it was very effective by itself, it's too costly to do what I want to. Alright, so I'll be using several methods throughout my research to test the microcidal effectiveness of my chemicals alone and in synergy coupled with each other. One method in particular that is very important for this research is called the Kirby-Bauer method. As you can see here, the Kirby-Bauer method takes these small paper discs, and you hydrate them with your chemical, and then you apply them to a culture plate that has Staph aureus bacteria smeared on it. So once you apply that to the culture plate and you have it incubate for 24 to 48 hours, you'll get these little clearings in the bacteria here, and those are called zones of inhibition, which what I'm looking at is which one of these acids, either standing alone or coupled together, will produce the greatest inhibition overall. Now, some problems arise with using organic fatty acids in that they are really, really sensitive, structurally speaking, and that sterilizing them was at first a problem. However, I found that I could vacuum sterilize these by filtering them through very fine filters to make sure that they do not contaminate the plates I'm applying them to. And as stated here, this falls under the purviews of aseptic technique, which is very important when you're working with bacteria, because if you want to get pure results, you have to make sure that there are no contaminations to your culture plates at all. And where the big series of measurements comes in is that I'm looking at 1 molar concentrations in micromolar quantities of standardized units of 10, so from 10 micromolar to 20 micromolar and so on and so forth, will provide the most optimum effect for Staph aureus growth. Next slide. And here to explain chemical synergy, I made this graph, whereas here on this axis we have drug Y by itself, and on this axis we have drug X. And normally, you would think that just adding two things together, they would have a purely additive effect. But what actually happens is that since the drugs interact specifically, they produce what is called a synergistic effect, which is a boosted effect overall, more than just a purely additive. So the way you can think of it is the sum of my parts are greater than my whole. And the broader impacts of doing this is that antibiotic use has become incredibly prevalent throughout hospitals worldwide, and using these, since they are not antibiotics and they're relatively inexpensive to make, I will effectively reduce evolutionary pressure that has made antibiotic-resistant bacteria worldwide. And it'll also be affordable because these acids can be produced in mass quantities. And here's my budget. I fell under budget, which is very good. The most expensive part is buying this because, well, this is the smallest quantity that I can seem to find. And I would like to give all my thanks to my mentors, Dr. Darren Bell for steering me in the right direction and making sure all lab safety protocols are measured, Dr. Duffy also for advising me and giving me advice on how to preserve aseptic technique, and Ms. Bergen-Losey for allocating the needed lab materials. I needed for this experiment. Thank you all. If you have any questions, you may ask them.